So this session will start with, so we will have presentation from the ended working group. There are 4. And so the first speaker, I will introduce the first one, because then she's also the host. So the first one is Poelchau. She will present about the achievements of the Data Federation Working Group. And her title is Data Sharing and Data Federation in Genetics, Genomics, and Breeding Databases, Current Status, Needs, and Future Direction.
Okay. Thanks, Marie-Angélique. Can you also make sure I stay on time?
Yes.
Since I'm talking and moderating. So hello, everyone. Hi, everyone online. Thank you for joining our afternoon session. So I'm going to start this off by speaking on behalf of the last year's Data Federation Working Group. We're going to talk about the progress that we made, the accomplishments that we had, and then we're also supposed to talk a bit about the challenges and sort of reflect a little bit on how the working group progress went. So I'll talk a bit about that. Before I start, I need to introduce our working group. So since I'm speaking on behalf of the entire group here. So we were a nice, well-rounded group of, around 12 people. Our chair is Jennifer Clark. She was here earlier over there in person, co-chair Andrew Farmer. And so we met. I wanted to give a little bit of history about this working group. I don't have a slide, so I'm going to ad hoc it, but when AgBioData got together in person for the first time in 2017, we actually had a similar working group that we started out with then called the Programmatic Access to Data Working Group, which was chaired by Ramona Wells. And at that time, that working group, kind of like most of the working groups for that session, had a number of specific recommendations for how to increase essentially making data more machine readable and programmatically accessible. So we had, as we started this working group, we had that to start with. Also in the interim, Meg Staton had chaired a Data Federation group, working group in the meantime that was sort of ad hoc when we were doing these unfunded working groups as well. And that was one of the reasons that when we started this grant, we actually thought it would be important to have this working group up and running. But once we got together and started reviewing what these groups had done before and thinking about what we want to do ourselves, we thought, it's been 6 years or so, or 5 years since this group got together, and we really thought we needed to have a fresh slate in terms of understanding the needs and use cases of this community in terms of data federation. And one of the problems that we also thought was, so we started already, and we already had the name coined Data Federation, but what does Data Federation actually mean? And as we were discussing that, we realized, in terms of thinking about access to data on a technical level, what is that? There's actually a spectrum, right? And if you look at this figure provided by Peter from our working group on the left side of the spectrum, we have less automation in our data sharing spectrum, where essentially we could just be mailing each other papers through mail, and that's sharing data, right? But it's not very programmatic at all. It's not programmatic at all, and it's also not very achievable in terms of having bigger, larger data sets and making that more automated across databases. As we get further along on the data sharing spectrum towards more automation, there's more technologies that we can use. An example is programmatic data access, where essentially you have data that you can access via code. Okay, so you can write a program to pull that data down in its metadata. And on the very right end of the spectrum, we have data federation, which is essentially a model where computer systems exchange data in the back end, but the user does not need to know where that data actually is. To the user, it appears as if it's one source. And so as we were discussing these different ideas in this whole data sharing spectrum, from no automation to federation, we asked ourselves a couple of questions that we addressed, hopefully, for the most part. So one of the questions we first had was, currently for AgBioData, what use cases for data sharing are there? So how are people sharing data? What are the problems? What's the current status of AgBioData on that spectrum? Where are we? More on the left, where we're mailing each other's files, hopefully not, or are we actually approaching a point where we can have that sort of data federation model? And then finally, if we get towards that more automated level of data sharing, it makes things easier in terms of moving big data around or accessing it programmatically. But there's also costs in terms of learning the technology and maintaining and developing it. So the question too is, so as databases, where do we actually need and want to be on that spectrum? Do we have to have that end goal as a federation, or where do we need to be? And so for this first point, I will talk a bit about just very briefly about our attempts to collect use cases. So we put up a GitHub site. You can see the URL here, a discussion group where we collected initially amongst ourselves use cases for data sharing in AgBioData, and during last year's online community workshop, we also solicited use cases. We had a hard time collecting them, so that was a worthy effort on our part, but it sort of fell flat. But if you have any use cases, please post them there, and we can still consider this as we move on with our working groups. Just note that. But for the second two questions, we decided to perform a survey, an assessment of the community in collaboration with the Ontologies working group, and I think we're going to hear about them in this session today as well. And we had 4 specific goals that we were trying to address with this assessment. So first, as I mentioned before, what's our current level of data sharing? So what are we doing? What data and metadata are being shared and how? The second question was, where do we want to go? What's the desired level of data sharing? And is there a discrepancy between existing and desired data sharing for databases and their stakeholders? The third question was, what are the barriers towards data sharing? And the fourth is technology awareness. So if we wanted to develop more automated technologies towards data sharing, what do we know about it? Is there a training need within the community for us to be more aware of technologies? So we decided to create this assessment, this survey. We created 20 questions aligned to those above goals, sent them out to all of you. Thank you for answering our survey, those that you did. In last July, we did for a month, and then we're working with the Ontologies working group to finalize this assessment. Today, I'm not going to show you all the results from this survey, but we are working on a draft manuscript publication to come out of this. So stay tuned for that for the full, if you want to have the full lowdown of that assessment. So in terms of results, so in terms of results for current levels of data sharing, one of the questions that we asked was, what data types do you share with other databases? We had 29 responses for this. And the gist of this is that by and large, it's very much the genetics and genomics aspect of the GGB, genetics, genomics, and breeding community. So most of us have annotation and raw sequence data, variant data, but there are, of course, some that have marker data, phenomic data, et cetera. And there are some other more unique data types here on this end of the spectrum. In terms of the mechanisms that we use for data sharing, so we had 29 responses for that as well. And we provided a list of certain ways to share data from, we have a hyperlink to a flat file, so you can click on the link and download it. Or we have web services where you could write a program to pull that data down if you wanted to. And so the bars here in green, the arrows in green are where I indicate, these are technologies that allow programmatic data sharing. And so the gist of this is that most of us do have some level of programmatic data access to our data. We did not tease apart how much of the data or metadata is actually accessible in that way. So that's something that we might want to follow up on later. And for respondents, for databases, do not use machine readable methods. So there is a bit of room for improvement there in that level. All right, so now where do we want to go? So here I have results for 3 related but separate questions, and I'll walk you through this one by one. So the first question was, how important is it to you, like as a database owner, curator, a database staff member, to make your database more discoverable and available? And you see those answers in blue here, where 1 is not important and 5 is very important. So you can see for this question, how important is it to us as databases? There's very much on the high end of the spectrum. We think it's important to make the data more discoverable and available. The second question was, how important is it to your user community, to your stakeholders, and from your perception of your stakeholders, to make your data more discoverable and available? And we see that in red. And it's similar, but not the same. So we seem to have a perception that our users don't think it is as important as we do. And then third, and in yellow here, how high is it in your development priorities? So are you actually going to spend money and time on making your data more discoverable and available? And things shift. We have here a mode of on level 4 of importance, but we definitely attribute are not going to provide as much funding or resources for development of data sharing methodologies or improvement of that than we assert its importance for. And that's sort of an interesting result, right? Is that we think it's important, but are we actually going to do something about it? Less so. I mean, we're going to try to tease it apart later. And so here's one of the reasons this might be happening. So here's our question on barriers to success. What do you feel are the biggest blockers to successful data sharing in the community? And this is not surprising, the results. So time and resources was the biggest one. Funding and return investment next. So these are things where a sustainability working group might help us out later. But two of the more interesting heavier hitters here were lack of data standards and technical knowledge. And there's a couple of other interesting use cases there, but they're lower frequency for us: institutional policy, data licensing issues, et cetera, sharing across borders. And our final group of questions was about technology awareness and training. So we asked, what data sharing technologies were you familiar with, which we see here in blue. So we provided a list of different types of technologies that we thought were relevant, from manual file transfer, hyperlinks, et cetera. And then we also asked what data sharing technologies would you appreciate learning more about, which is here in red. And so what I've circled here with this black box is essentially the sweet spot where it seems like we have people who would actually want a critical mass of AgBioData where there's a bit of a knowledge gap, and we might be able to address with additional training. So based on these results, we provided a couple of recommendations for AgBioData, some of which we're able to act on actually. So the first is identifying solutions to funding problems. So that was one of the main barriers to data sharing is money. And so we're hopeful with addressing the sustainability working group here that this might help provide solutions to funding problems for databases that would provide means to improve data sharing on a technical level. The second point is data sharing and training for database personnel. So we had that last graph that I showed you that had the specific training types where there might be knowledge gaps. And we also know that for 47% of the respondents, technological knowledge was a substantial barrier to data sharing. So we actually started a new working group based on the results from the survey that we're going to hear about tomorrow. Peter's going to tell us about that working group. He's the chair of that working group as well. So we have a direct consequence of this survey in a new working group where we're going to try to identify and provide training on specific data federation and data sharing technologies. The third impact here, the third recommendation is, we think that one of the reasons that there's a discrepancy between the importance that we assert, the importance that we assert for data sharing or improving it, and the user stakeholder community is that and our development priorities is that we might want to educate the stakeholder community more on the importance. And I'm out of time. So I think the last thing I want to say is okay, thank you. And so one of the things that I did not have time to talk about in the survey results is that there is a need for a better understanding of the problems around sharing phenotypic data. From this working group, I didn't show you the results, and also from some of the working group suggestions from this particular year. So in September, I think, stay tuned, we are trying to hold a webinar session, discussion session online on where AgBioData we should come together and find the most low-hanging fruit or the most salient problems for us in terms of phenotypic data sharing. And we also need to continue work on standards improvement, which I learned today that the standards for genetic variation working group is also thinking about that for that particular data type. We had some challenges, so we were asked to comment on these challenges as well. So I think as many other groups had, we had challenges with time zones and internet connectivity for certain, we were distributed pretty far at first. As I mentioned, we had to spend some time deciding on the working group focus based on what the previous groups had done before us. getting use cases was a problem. That was a bit surprising to us, but here we are. And then developing a shared vocabulary. So for example, we started out thinking about data federation, and then we broadened it to a broader sharing concepts. What went well? So it was a fun group of people. We had some really good discussions. We really appreciated and enjoyed the community workshop last year. We got some really great feedback back there. We were able to work together with another working group, the Ontologies working group that we'll hear from later on the survey and later the manuscripts. And we created a new working group based on the feedback that we received. We were asked to have some specific questions for the community. So here are a couple if there's time. We wanted to know if there are other communities or groups outside of the AgBioData working on data sharing technologies that we should be aware of. For which types of data does our community have the most urgent need for standards, since that was identified as a blocker? And which use cases would provide the most compelling demonstrations of the benefits of data federation? So that was really one of the things that we were thinking about is if we want to actually improve the adoption of newer technologies and incentivize that, you have to know why you're doing it as a PI to allot the resources to your project to that particular development. And so compelling use cases help with that. So this is all I have. If we have time for discussion of those questions or any other ones, I'm happy to take them.
Are there any questions for Monica? Question. No? Okay. So I can move to the next.
Moving right along. Okay. So next up, we have Jung from the Genotype-to-Phenotype.
Can you give the updates for the federation for some of these standards? How much do you guys look into like things like what humans have for the project that allows them to identify data sets? So like the human projects, especially the clinical data, have quite a few standards now out in G4H on standards, and some of the standards are about interoperability and how to identify data sets. I'm curious if anybody has looked at that.
Yeah, no, we didn't really delve into standards per se. We just identified that as a blocker, and so that would be good if you have. Yeah, if you could refer like the steering committee to those actually, and then as we consider new working groups for next year, maybe we could set up some feelers to see if there's interest in adopting any of those, having a working group on that. All right, so we have Sook coming up next, who will talk to us about genotype-to-phenotype working group.
Okay, so the title of my presentation today is Agricultural Sciences in the Big Data Era. Genotype and phenotype data, standardization, utilization, and integration. Pretty long. So I'm representing the genotype-phenotype working group. And these are our people. And so as you can see, we are pretty diverse group of people. I think we are representing 7 different institutions in 7 different countries. And if you think about the countries that we are originally from, I think it's most like 11 countries or something like that. And also career-wise, we had graduate students, postdocs, and like more senior professionals, and also bench researchers, data curators, and bioinformaticists. So it was like we were pretty diverse, and we had a lot of fun. So the overview of the presentation, so I'm going to talk about the aims of our group and then the accomplishment, more detail like what and how, and the recommendations that we came up, and then the challenges and what went well in our working group. So, so the goals were to improve the data collection and the data sharing, to facilitate linking genotype and phenotype data, and to promote data interoperability and reuse. So it was such a big goal for us to achieve. So we spent quite a lot of time, like what types of data are we even talking about? So like genotype and phenotype. So here, for genotype, you have whole genome, epigenome, genetic variation, QS, QPL, and the genes. You know, and then for phenotype data, you have the traditionally like these quantitative and qualitative trait values, and also you have using phenotype and now more like more advanced genomics data, but then you also have these molecular phenotypes, like transcriptomes are also phenotype, and proteome and metabolome. So we had a lot of data to cover. So we were like, so what can we do? So we were like, so let's find out the current status. So current status meaning like where the data is stored and what data and metadata are kept. And how are they currently integrated and how can they be reused? What are the limitations for reuse? And so if we find these out, maybe we could come up with recommendations. That's what we were thinking. And then we thought maybe if we do all this, we could write a white paper. So we did. So, like we submitted this manuscript in April 2023. So we started like last year, the year before, right, 2021, right? And so we really wanted to finish by December 2022, but then like writing process and everything goes on, and so. But I'm just so glad that we were able to do this, at least this April. So this is the diagram that we made for the introduction part. So potential integrative analysis using genotype and phenotype data. So like if all the data are like integrated and reusable, like then like the genotype data, like whole genome sequences, transcriptome, epigenome, molecular markers, combined with phenotypic data like traditional trait measures. I already talked about the types. So if these can be integrated and reused, like we could do GWAS or meta-analysis and comparative genomics, gene expression analysis, functional pathways, like a lot of good stuff actually. And so GWAS and QTL is kind of like it's analysis that you can use, you can do using genotype and phenotype data like meta-analysis, but also GWAS and QTL also produces data that can be further integrated, right? So, so like here, the little icon says the limitations. So that is like the data size, or this is due to data heterogeneity or metadata, the lack of metadata standardization, lack of infrastructure. So each data type, there are these limitations that we identified in the beginning. So the strategy for the white paper was like, obviously we need to have introduction, main content, conclusion. But then for main content, when we started writing about it, we talked about like we need to cover all of these. So we had like diverse enough people who wanted to focus on each of these data types. So we had whole genome, transcriptome. So in the earlier slide, I said like transcriptome goes to phenotype, but then the data structure wise, it's better to go with genome data. So we put them together, and then we have genetic variation, phenotype, genomics, proteome, metabolomics, and GWAS and QTL. So for each of these data types, we were planning to find all of these bullet points. So we started writing a brief introduction and the data type and method, and where are the data submitted, what metadata and data are submitted, and are there minimum standards, what type of studies we use this data, and then what are the limitations. So, but then, so we wrote all of this, and it was like huge documents, like too long, and who's even going to read this? And so we decided to combine them. So like, the first part, like genome transcriptome, genetic variation goes together, and all the phenotype data together. And, it was Sushima's idea. And so like a GWAS 2TL, so we, combine them together. And then, so for each of the, sections, we wrote this. And then the limitations, we put them all together because the limitations were very similar for each of the data types. So the final structure was this, like introduction, genomics, transcriptomes, and phenotypes, and GWAS, and then we had limitations and challenges and recommendations. So for the genomics and transcriptomes part, so we had introduction on technologies, the sequence and genotype data, and then the public repositories and uses and applications. So public repositories, like it was pretty obvious to have like primary databases, but then we realized a lot of top and class community GGB databases, they also collect the whole genome data and genotype data and transcriptome data. Also, they collect all kinds of other types of data as well. So we had the first table, so it listed all the primary public repositories. So we made it really simple for the table first, but then we had two supplementary tables to have all the metadata and all information. And then, so we, as I mentioned, so there is a crop community GGB databases. They have really like they focus on data integration for like whole genome, transcriptome, genotype data, either from direct submission and curation and or from the primary leads. So and then these data get integrated with all the other types of data like QTL, GWAS, phenotype, germplasm. And so these type of data are mainly by direct submission and curation from publications. So we made a table of all these databases. Oh, I forgot to say, so in the beginning we said we need to somehow focus. We cover all of these data types, but we wanted to focus on plant because we just couldn't do everything, and we had no one who had expertise in animal data. So it covers most of the AgBioData member databases, but then only plants, and then the databases that collect public, right? I think we had some categories, so public genotype and phenotype databases. So those are here. And supplementary table 3 lists all of these databases with more data like metadata and submission format and the URL for the data submission. So for phenotypes and phenomics, so we again had similar structures like data types, repositories, and knowledge bases. And so for these trait, phenomics, proteomics, and metabolomics data, and then we had separate section for GWAS and QTL, and then we had formats, standards, and metadata. But then for all of these, we made one table. It's huge. I don't expect you to read, but we had like different sections in the table. So we had species, distribution collections, and traits in QTL, and phenomics, gene expression, proteomes, metabolomes, and secondary knowledge bases that combines these phenotype data together. So then we had data reusability limitations and challenges. I think we've talked about this all day today, basically. So but then we figured there are really 3 categories associated with it. So challenges regarding the data. And then the second is resources and funding. And third, implementation of fair data policies. How do you implement? We all know this, but so data wise is like the diversity and format heterogeneity, size and object identification or lack of it for metadata, So these are all the challenges, but then also important is resources and implementation. So the recommendations, we had more than this, but I just tried to focus on important things. So we really need to somehow standardize the data collection and protocols and consistent data annotation and data quality control. And also we need like data storage infrastructure and data management software and curation tools for that is to happen. And then the most importantly, concerted effort, like what we are doing right now, to make multi-omics data sets interoperable. So we, as a consortium, I think we are starting and like doing an important role in this. So I just wanted to have this, we all know this, but then how, I don't know how correct this is, but then, like curator time is basically the same. But then if you think about the data volume and complexity, it's huge. And then someone needs to realize it. Yeah, so what's really needed is the sustainability of the core databases again. Okay, so what challenges our working group faced is very it's similar to with other working groups, I guess. But then so the goals were too big, and we thought like it's just something that we cannot. So we tried to narrow down the scope or try to find what we can do. And also the multiple time zones, we had like people from New Zealand, Europe, and it was pretty difficult. And everyone is already busy, so we need to divide the work and try to be flexible. So what went well? So like we like having wonderful people and who really wanted to get work done. So that was really helpful. And then a regular meeting at a fixed time. So I tried to find out how often we met. So in the beginning we had bi-weekly meeting, but then like since August to 2022, I think we had weekly meeting until we submitted the paper. So and then I think like regular meeting reminders, like we met on Monday and like we tried to send reminders on Friday and Monday morning and it worked. Also like having a finished product white paper, it motivated people a lot. So finally, acknowledgement, the steering committee members, and we also have past steering committee members, past PC, and EAC members, and also NSF for funding our work.
We have plenty of time for questions, though. Go right ahead.
So like you said that you have to narrow down the scope. Is there anything that you didn't cover and you think it would be important to cover in the future?
I guess we did the white paper, but then we didn't really like come up with recommendation or some. I mean, I think there are a lot of work to do. Maybe you can even narrow down further and then do some pilot study or something, or like go wider and include animal data types. So,
yeah. I mean, one thing we did not do was to which data can be integrated together, which data types and which cannot be. And, so we try to cover two platforms, that is genome browser and pathway browsers, and what different type of the data can be incorporated or integrated there. But we did not cover a very detailed aspect of heterogeneity of data, how to put that together, and recommendations. So that is a huge step.
It's okay, I'll speak loud. So I'm curious, the first group was talking about sustainability. With your white paper, did you actually make recommendations that could be utilized to support, for instance, funding initiatives to support some of the standards for support the scaling, or things that are currently challenges as part of that white paper?
So the recommendations I shared is part of the recommendations that we wrote in the white paper.
So have you shared those with the funders as of yet?
Not yet.
Share those with the funders?
Yes.
We thought we share the paper when paper comes out.
Yeah.
Don't wait. No, seriously.
Yeah.
Don't wait for the paper to come out.
Yeah.
File
It's right here.
the print server, share it with them now.
That's actually good. That's a really good point about like just a principle for like the products from AgBioData is, if we put things out in like bioarchive, then it gets it out there for public comment. So it's a way of incorporating community feedback. Like, we can say, hey, we have this thing out here, what do you think about it? And then I guess, send it to our beloved program officers.
Yes.
Yeah, I like that idea.
And possibly as part of the parascientific literature, if we move on to also include funders, not just publishers. That could be a conduit. We have time for another question or two. Marie?
Just a comment. I think you guys did an amazing job. But I think you should actually really calculate the amount of hours that went into it. So that when you do send it to the funders, you clearly articulate the investment that went into generating it. It wasn't just like a one-off.
And voluntary effort by all the contributors, nobody was paid.
Yes.
I had a hopefully quick question, but maybe not so quick. So you had the phenotypic data types categorized, and we're trying to think, ahead about phenotypes and where the problems are and where we as AgBioData might have some impact in terms of improving standards, data sharing, et cetera. Was there any data type that stood out in your review as one that we might want to focus on for?
I'm not sure, but
I think images.
Images.
I think images of different phenotypes, and there is no public repository or there is no curation of annotated images. That was big thing, and I think the phenomics data, that is a lot of it is image-based data. Some of it is the colorimetric and chlorophyll fluorescence-based data. So
if I add to it, like so phenotype data that we had repositories, but then like for crop phenotype data, I think it's just like we don't have repositories except the GGB data. Like for crop specific.
Yeah.
So it's just like crop community databases is the only place. Like for genotypes, you have EVA and stuff like that, and sequences you have NCBI and all that. But then phenotype data we just don't for crop. Crop specific.
All right, we need to move on. Discuss it. Thank you. So next up, we have Marie-Angélique Laporte, who is going to be joining us online. We need to stop sharing. And there?
Yeah, I'm here. Can you hear me? My connection is not so good. I'm sorry.
We'll bear with you.
Can you hear me?
Can you hear us?
Yes.
Great. Can you share your screen?
Sure.
And the screen that helps with the connection.
Yeah, if it helps, I can share your slides and advance them for you, if you prefer.
Yeah, maybe that's better, please. Yeah. Because I don't even dare to move or anything, like, you know. I'm so sorry. Yeah.
Great. Slideshow. All right, can you see this, Marie-Angélique?
Yeah, thank you. So yeah,
All right.
so I'm gonna present. Can I start? Can you hear me? Yeah.
Yes, we can hear you. You can go right ahead.
Yeah, okay. So I'm gonna start to present like the work that we've been doing for, in the ontology working group, and then I'm presenting also on behalf of Laurel Cooper, that was the chair of the co-chair of this working group with me. So next slide, please. So yeah, so that just an overview of the presentation. I'm just gonna introduce what are ontologies in the context of the working group, because I think that's important. I'll do that very quickly. Then I will present some slide on our working group and who were the member of the working group, then some results, and finally the challenges that we faced. Next slide. So the working group was called like ontology working group, but really, I mean, we didn't discriminate in terms of what are ontologies. I mean, in our context, it can be anything from a glossary to a really formal ontology. And so really, depending on the database or depending on the topic, I mean, we've seen a lot of users also having their own control list of terms. I mean, we accept that as being ontology, and we think it's already important to be able to describe what are the data and how that they are used and what they are really are. So everything in our context is an ontology. Next slide. So a bit about like our working group. So we started meeting in November 2021. We had bi-weekly meetings until the end of November. I mean, the group size varied depending on the meeting, mainly because, I mean, we were like, you know, spanned around the globe. So there were people from New Zealand, from Europe, from North America. So really, I mean, that was a bit challenging at some point to find like a time to meet and all of that. But then, I mean, it worked well at the end. Then, yeah, the attendance was about 6 members per meeting. And but then also something that needed to be noted is some people register to be part of the working group, but at the end never really participated. Next slide, please. So the next slide is the core members of our working group. So the people that participated to the discussion and help us build like the result that we had. So yeah, I didn't list the affiliation, but yeah, really it was from different types of institution. Next slide, please. Previous slide, please.
Oh, sorry.
Yeah, thank you. So the objective of the working group at first was really to have like a survey to better understand like how the use of ontology evolved from the survey that we did in the previous phase of AgBioData in 2017, and what it is now, to see if there was like, you know, things are changed. One objective of our working group was also to identify the tools that were missing that might be helping using ontologies more easily. Another goal was really to identify the barriers to using ontologies. Then we also had the objective to invite, like organize like a webinar. Ontologies, how they are used and how they, just to show example or show by example how this can be done. And the long-term goal was really to set to have like a recommendation, of course, and to have a paper. So that was the plan. Now I'm going to present what happened during our meetings. So next slide. So what we accomplished, so we were able to do a survey in collaboration with the Data Federation Working Group to really targeting like the AgBioData community. We have like a paper that is on preparation with the Data Federation Working Group. We were able to discuss and identify a list of use cases for ontologies in the community to identify like the gaps and barriers to the implementation of using ontologies in the databases. And then we were able also to organize a webinar on ontologies and fair data that were done by Chris Manga. Next slide. Now I'm going to explain a bit more what we have. So first, the survey, I mean, the Data Federation Working Group already presented what we did, but yeah, so we conducted a survey using Google Form that went out in July 2022, and our goal as the Ontology Working Group was to better understand which ontology are used by which database and to annotate what type of data. So the survey targeted only the AgBioData community because we really wanted to focus our recommendation on the community. And so we received responses from 33 individuals covering 37 databases and resources. But a lot of response was skewed towards plant researchers, like 24 out of the 33. Next slide, please. So here it's just like, so the tree graph. So we decided to analyze the data by type of database. So the plant database, the animal database, and then the databases that were not really plant or not really animals, or that were a mix of the two. And so we can clearly see that the gene ontology and NCBI taxonomies are the ontologies that are used across all these different types of databases. And then depending on the communities, then there would be more specific ontologies. So some are only plant specific, some are only animal specific. But also what we realize is like 30% of the respondents mentioned that they were not using like well-published or not well-published, I mean, open ontologies, but they were more using like, you know, their own in-house ontologies or vocabulary. So that shows that even if people or in the community don't have the resources to use ontologies, I mean, they can still start this work of standardization by working on their own term. So next slide, please. The other key results from the survey were that a lot of the people in the communities are not creators of ontologies, they are just users of the ontology. I mean, they don't manage any ontologies, they contribute often terms to the ontology, but not really maintain them. And so a key point or a key factor of contributing to this ontology was through GitHub. So that really helped the community to better contribute to existing ontologies. Also, the survey highlighted that ontologies are mainly used for annotation, curation, and for search archiving. That's the main two use case where people are using ontologies. I mean, they are not used to do fancy knowledge graphs or, you know, derive new knowledge or things like that. No, just curation. I mean, just that's already good, very good. Annotation and then search improvement. And then the main barriers to the use of ontologies were insufficient funding and expertise to get started. So it really shows that, you know, yeah, more investment is needed, but also probably more training as well. And also one key barrier was also that often ontologies don't really fit the need as they are, because there are either some terms or some key concepts are lacking, or sometimes they are way too specific. So that's also like a good feedback for the people maintaining the ontologies. Next slide, please. So as I said, we also discussed many use cases. And we tried also to understand for each use case what were the gap and the barriers to these use cases. I'm not going to go into details, but I mean, I think Sujama mentioned just before, like the high throughput phenotyping. I mean, we discussed that quite a bit also, because we think that there is a big gap in the ontology world in terms of terms that, how this type of data could be described using ontologies. So really, a big gap will be that new parameters will need to be created. Next slide. So yeah, you can, I mean, yeah, you will see the slide, and that's also like more use cases. Like, and again, I mean, if I take just one, I mean, if we look at, graph use of the ontologies, that will require to combine different ontologies. Also, this is not easy, because I mean, a lot of ontologies have different scope and are using different standards, let's say. So it's not easy to combine everything and all the data together, even if we have ontologies. I mean, maybe they are not talking to each other enough. Next slide. Then, yeah, just a quick summary. I mean, just to remind you that, so there was the interesting webinar by Chris Mungal. We explained like the entire history of the gene ontology, but also something that he presented during his webinar was all the tools that the Obo Foundry community are developing, and that really people can build, can reuse easily, and that can really facilitate the use and the maintenance of the ontologies. So really a lot is happening in this Obo Foundry community, a lot is moving. So I think it's really key to keep up to date also to that community to better use ontologies. Next slide. And now this is like the challenges that we face. So we also had an issue with like the attendance of the members, but I mean, as it was said before, I mean, it's mainly because we were stretched across different time zones. So at some point it was challenging, but I think at the end it worked better. And also, I mean, we wanted, so we identified like all the different use cases. We discussed some of them in depth, but at the end, I mean, we couldn't really implement any of these use cases because, I mean, like that would require a bigger time commitment from our members. So yes, and also, I mean, one of the challenge is like we also shift a bit from our objective that we, and everything that we wanted to achieve in this working group to what we really achieve at the end. So yes, and I don't have a slide, but on the things that went well, I mean, also we had great discussion. I mean, it was very interesting to discuss. I mean, for me, who worked mainly on plants, to discuss more about what is happening in the animal community. I mean, that was also really interesting. And also like the different people, the way they are using ontology differently, that was also like a key highlight of our working group. And that's all I have for today. Thank you.
All right, thank you, Marie-Angélique. I think our microphone is dead, so maybe the audience members can ask questions and I can speak them into the microphone here so you can hear them and respond, or maybe. So, and we have time for just a few questions. Yeah. I can start off with one, Marie-Angélique. So I noticed that you were able to collect a fair amount of use cases. We had a pretty long table. And in the data federation work group, we had a hard time actually getting them out of people. So how did you manage to get so many?
I don't know. We just discussed, as I said, I mean, our group was quite diverse and we have different interests. And so that was just like, just list all the use cases that you can think of. And that's what we started discussing it.
Great. Lenore has a question.
Yeah, I was thinking about with ontologies, right, that they're often not really static kinds of things, right? So when thinking about like the ontologies that we need, did you have any sort of thoughts about like ones that potentially needed to sort of grow and develop? I know you said that there, like you know, everybody's using the gene ontology and that's really well supported, but, you know, are there other ontologies that seem really critical, particularly for like the phenotype space that really need attention?
Did you hear that, Marie-Angélique?
Yeah. That. Yeah. I mean, I'm just thinking, like, you know, I mean, it's late, so forgive me. But yeah. So yeah, I think, I mean, all the ontology, as you said, I mean, ontologies are changing. I think with new topics and new research areas, I mean, ontologies need to evolve. But I mean, when you need to, I mean, asking ontology maintainers. Maintaining an ontology, it's easy as long as we have access to researcher that could provide the terms. So I think, I mean, having these new ontologies or new part of ontologies, like, I mean, that would require also to get feedback from the community to the people maintaining and creating these ontologies. And so GitHub, I think, is the good way to start. But if something is totally new, it don't fit into any existing ontologies, then yes, I mean, I don't know how to do that, but yeah, a conversation will need to happen with someone who would be willing to create a new ontology.
Think a lot about like the single cell data, you know, just do we really have a robust ontology for describing all of those cell types?
Cell types, image types, a lot of like phenomics, high throughput data.
Yeah, definitely. All the high throughput phenotyping data, I think that's a bit missing currently. So yeah, so that will be something that will need to be worked on.
Still have time for one more question.
I don't have a question, but I wanted to acknowledge Laurel Cooper's contribution to this working group. She put a lot of effort into putting the survey together and doing some editing, and she's not here today, but she did a lot of work.
Yeah, she's all right. Yeah. Here, in case you couldn't hear that, acknowledgement of Laurel Cooper's contributions to this working group.
Yeah, definitely. That's what I said. I mean, she was the chair. I mean, I'm just co-chair because of, yeah. but yeah, she was a chair. I mean, we were co-chair, but I think she was more chair than I was. Yeah, but she can.
Right. I don't know if there are any online questions, but we might just move on. So thank you, Marie-Angélique.
Thank you.
That worked out well. Okay, and so next up, we have another online speaker. Stop sharing this. Next up, we have Gerard Lazo, who is going to speak to us on behalf of the Pan-genomes working group with the title "Running the Spigot of Genomes into the Pan Era. How should we pool data?" So Gerard, can you say something so we make sure we can hear you?
Yes, can you hear me okay?
Loud and clear. Excellent. And
Good.
you're driving on your own, so I will leave it up to you.
Okay, very good. Great. Well, thank you for having me speak. Let me be part of this. Yeah, so our working group it was started out at the beginning in January of 2021. However, our last real meeting was basically in September of 22. We've actually had a bit of scheduling issues, but you know, some of the past and you know, members and participating members of the working group are listed in this slide. You know, most of the group worked on plant genomes, and let's see. Well, most of them were plant genomes, but others were interested in just the general issues associated with pan-genomes. And then what I hope, and then throughout this time, we've had a few presentations, and then we had a lot of scheduling issues where we weren't really able to meet on a consistent basis. But just to give a brief background, you know, when you consider pan-genomes, everything starts out with its first reference genome. You know, so and when you're looking at the pan-genome perspective of things, you know, it could range anywhere from looking at viruses all the way to, you know, your larger eukaryotic organisms. And you know, most of these genomes, like for instance, for the viruses were very early, but most of the sequencing didn't take off until the turn of the century. And in my case, I work mainly with the cereals group, which would include the wheat, barley, rye, and oat. This is in association with the Grain Genes Project. And one of the very first things that sort of introduced me to pangenome concepts was an early paper before all this genome sequencing started to take place. It was mainly based on a collection of molecular markers and a lot of genetic studies. And Catherine Devos and Mike Gale back in 1997 posed this view of the cereals in general, ranging all the way, from rice up to the larger genomes of the Triticale. And then, this was back in 97, and so up to now, most of these genomes have sequence, and they have sequences or reference genome sequences available, and in some cases, many times over that. And so in my case, for instance, to take example for wheat, for bread wheat, which is the hexaploid genome, which has the A, the B, and the D genome, there's quite a collection of pseudomolecules that have been generated. Not all of the molecules are considered, of the reference type standard, but, the very first genome that was done for wheat was the Triticum estivum Chinese spring cultivar, and that was done back in 2018. But there was a lot of information that led up to the release of that genome sequence by the International Wheat Genome Sequencing Consortium. And then since that time, there's been additional sequencing to refine the Chinese spring genome into a reference standard genome. But then again, you need to consider that these genomes, for bread wheat, each genome is about 16 gigabases in content, and that's just the genome itself. And then you add on all the metadata that goes with it, it starts to exponentially grow. And as part of the Grain Genes team, we collect this data, and the Grain Genes is a public resource where we collect a lot of the public information and try to distribute it in a way that it can be used. And through the Grain Genes project, we have a lot of markers and genetic information that's attached to the genome, but we also show this information through genome browsers, through blast searches, and also some map displays using the CMAP interface. But one of the things, when you're trying to move more into what you would consider pangenome, surveying, all the genes, all the genomes across all the different references that can be collected, this can put a strain on some of the compute resources that you use. And, for instance, here, currently at Grain Genes, we have quite a few of the wheat genome pseudomolecules. We have the works that were published for barley from several different germplasm. And then there's also some upcoming data from oat that will be released. Also, as another part, we use these BLAST databases, and we use a modern version of this BLAST database called Sequence Server. And it's been very helpful in being able to sort of select and pick the genomes that you want to focus on, and then try to do blast searches against those genomes. But again, you have to consider, for the wheat genomes, it's like 16 gigabases of data. For the barley genome, it's about 6,000 gigabases. And then for oat, it's also about the same. It's about 10, 12, 14 gigabases in size. And this can put a constraint on some of the compute resources. So some of the things that we're trying to do in trying to manage some of the pangenome data that's coming about is try to optimize the indexes that are used to search some of these BLAST databases. And in the example here, I just show 4 different ways of searching 3 different cultivars. So in the upper left panel, you have the entire genome. So if you had, and you're assuming about 7 gigabases per chromosome, so if you had for 3 times 21, you're going to have the that's not right. Well, let me get the note. Yeah. So you're going to have about 44 gigabases for the entire genome. And then if you were to focus on each genome group, there's A, B, and D genomes, then you'd be looking at 14, or about 15 gigabases. And then another way of looking at the data would be to search across a single chromosome, but each of the different genomes that's represented. And then that would be like 6, a little over 6 gigabases. And then if you were just going to focus on one chromosome and one group at a time, it's about 2.1 gigabases. So in trying to survey in a pangenome perspective, this is only 3 cultivars, but considering we're easily going up to like 50 different genomes to search at a time, so some of these these represents amounts of memory on the machine that are being used, and it's best to try to optimize your systems in order to get the data in an on-demand fashion faster. And so one of the ways that we're taking some of this data is we're sort of grouping it into the groups. So, so if we have 30 different genomes that have been sequenced, so now when you're looking at group 1, you're looking at the, 30 times, you're looking at 30 genomes. And in this case, it'd be group 1 times 3. So it'd be, 30 of the A, 30 of the B, 30 of the D genomes. And in most cases, because of the organization of the genome, the nomenclature for each one of the chromosomes, they're very synthetic in their design and nomenclature. So group 1A is very closely related to group 1B and group 1D. And so you would be able to see the slight changes in the genomes as you would survey those. And then at the bottom, we also include the reference genome for Chinese Spring to it as a comparison. Other genomes, like, right now there's about 30 wheat genomes available. That, let's say, for instance, now if you look at some of the other databases, there's, in the rice world, there's about 251 rice accessions available for searching, and this is just an example of the site for the pangenome information for these accessions. And then again, there's also the rice, the Gramming group also, I believe, interacts with this group and also has other pangenome type representations within their displays. But another aspect of considering the pangenomes, is the visualization. Doing blast searches, looking at maps. It can be quite complex, and so why and in surveying our group, because we had people that worked with viruses all the way up to higher eukaryotes. The needs for pangenome or the expectations from pangenome analysis can vary considerably, and so that was sort of an issue that we were sort of grappling with in our group. But let's say, for instance, for the wheat or cereals or plants, why would you look at the pangenome analyses? You want to look for evolutionary studies. you want to look for genetic variation, identification of introgressions, et cetera. Also to study important gene families, and then also to use it for ultimately, try to use this knowledge for crop improvement. That's something that is highly desirable with our changing environment. But the existing resources, sometimes they provide nice overviews, but sometimes they're too complex. And there's always a number of new tools that are being developed, and there's always being continued refinements. And you want to be sure, or you would always like to have the latest reference quality sequence available to you to do these studies. And in most cases, a lot of these displays are not instantly publicly available. A lot of times data has to be downloaded, and you actually have to do a lot of the analyses, off site. And so, and then there was a publication that came out recently that gave a nice review of a number of pan-genome tools and recent studies that have been done. And then there were also some GitHub sites that were identified that sort of pointed to some nice tools that are up and coming and on their way and have been tested. But all this software that's being developed, needs, it's being tested, but it needs a resource to put it to the test. And for instance, the one displayed here is a gene space, which there's an image showing alignment of all the serial genomes, and then Panache, which is another display software that was first used for banana, and has also been applied to wheat as well. And then there's a number of tools that have been classically used in a number of databases. For instance, JBrowse and AccuSyn, and actually the display that I'm showing here is sort of built off of the Circos. It's more the early Circos display. And then on the left, the JBrowse display, it's been improved enough to allow a good comparison or comparative analyses of a lot of within a genome browser. And in the example shown here is a diploid line with 7 chromosomes compared to the hexaploid line with 21 chromosomes. And then again, there's which visualization tool you want to use and how do you want it to display information about your pan-genome analysis. And all I'm showing here is there was just quite an array of different tools that were looked at within the group and discussed. And in most cases, a lot of people say, it's nice to use, but how do I get to using it? And that's sort of the hurdle in all cases. There's a lot of nice tools out there, but there really needs to be a sort of a faster way for someone to jump into the game to do a pan-genome type analysis. And so some of the I guess in our group, because and also because we had a drought in some of the meetings that we were able to make, we had some challenges. We had a lot of time challenges. There were also a lot of organizational issues that occurred, and everyone in the group was motivated, but the time seemed to be limiting as we went on, and many times topics came up where the project needed interactions with some of the other groups, and I think a lot of, and what I was trying to listen to in some of the earlier presentations, was there any discussion of pan-genomes within those groups? Actually, I was told that there was, but I didn't really hear that much mentioned about pan-genomes. So, it sort of may be a independent project utilization of this type of information. One of the things that we can do, some of the goals that we had tried to discuss was, trying to include, as I was showing examples of the visualization tools that are available, discussion of file formats with, how would you organize a pan-genome database, and a lot of that would have to be coordinated with some of the other groups. And then there was a brief attempt to try to build some tutorials on, how would I start in beginning a pan-genome analysis. And then again, with the nomenclature and there were some debates about, what are the real decisions or real definitions for some of the terms that we used in pan-genomes. And I think that mainly had to do with the different approaches and different utility that the pan-genome information would use for each group. There were some things brought up earlier about like the data federation. I think, there could be better ways of housing that data, and trying to making it more accessible to groups, rather than trying to start from scratch and build a resource to do the studies that you want to do, or to have the servers that provide the information have easier ways to have data downloaded to the consumer to analyze the data. There's been a number of these approaches done, and I think the role of what the pan-genome group can do, I think this is something that could be discussed probably in your breakout or your following session, just to discuss, what's a good way, what's a better way, should we do what we're doing now, or is there something that we should change? We probably need a little bit of a reignition of our efforts within the group, and I think we're willing to hear feedback from the community. Thank you.
All right, thank you, Gerard. We've got time for 2 questions. Tanya.
I'm wondering if he has a favorite visualization tool, and of all the ones he showed.
Did you hear that, Gerard?
Do I have a favorite?
Yeah.
Oh, well, right now the ones that I'm focusing on are using the JBrowse tool, and then the AccuSyn. That one is a tool that's connected to the database more intimately. So you can go to the database, you can go directly to the JBrowse display, or you can go to the blast search and then go directly to the JBrowse display. And then taking a lot of the pre-computed alignments between the different genomes, we're able to create the Synteny displays that were shown earlier. And there's a lot of plugins that are being added to the JBrowse 2 environment that hopefully will extend the ability to do studies a little bit more in detail. The one that I showed was more of an overview shot, but you can actually focus in within the key regions of interest to extract out information that you may need.
Thank you. We are basically at 10 till, right? So we're gonna wrap things up. Thank you so much, Gerard. We can continue discussions online if there are more questions. So let's thank our speakers.